Okay, could somebody confirm that you can hear me?
Yes.
Great, thank you. Let me share my screen and we'll begin. Let me start, before we begin, are there any questions? I assume there's no questions, so let's begin. Oh, that's the wrong Zoom part. That's making the, there we go. You should see my screen now. All right, today is October 11th, and we're supposed to be talking about Lab 5 today. Lab 5 is Streaking for Isolation and Culture Media. Please let me note that you should read the Unknown Project instructions file on Canvas for next week, because next week we're going to discuss the Unknown Project. And I don't know if I've got that. Yeah, I don't have it. Well, let's go there. I'll show you where that file is. It's under, I think, week 4 or week 5 of the lecture. My computer is kind of slow because the Zoom program is making the lecture lesson file. You can see how slow this is. Zoom used to be finished in 10 or 15 minutes, but they had an upgrade and now it's over 20 minutes. I think it's about 25 minutes to convert the lesson file. It converts it into a format that I can put it up on YouTube. Initially, I think it's just a Zoom meeting file, which isn't anything anybody can do. Nobody can use it except for Zoom. Can you see my screen?
Yep.
All right, I've got Read the Unknown Project for next week. And there's the Unknown Project. It's under week 5 of lab. The Unknown Instructions is what you need to read. I'll be talking about it, but it'll be helpful if you have some idea what I'm going to be talking about, because a large number of students, maybe a quarter, maybe even sometimes a third of the students, they never understand the Unknown Project. It's a project we're going to be doing on the computer, but it's going to be similar to you working in the lab to try and identify a bacterial species. It's just everything will be done on the computer. Any questions about any of that? We'll talk more about the Unknown Project next week, but do read that before next week. All right, let's talk about Lab 5, Streaking for Isolation in Culture Media. Let me do mention that I did grade labs 3 and 4 this weekend. And if you didn't get a perfect score, 100%, you can resubmit your worksheet by Saturday for full credit. All right, lab 5, culture of bacteria, culture media, inoculation, and isolation. There is some reading to do, but not very much. What is that? 5 pages there, and then 1 page here. And then there will be some video clips today and maybe a virtual lab. I don't remember if this virtual lab uses Flash or not. Let me see what this one is talking about. Media, cultivation of bacteria. I don't remember. All right, your learning objectives. Upon successful completion of this lab, you should be able to, 1, understand the importance of proper specimen collection and transport to the laboratory. 2, be able to explain the 5 principal steps used by microbiologists to process specimens once they're received in the lab. 3, be able to differentiate between various types of culture media. 4, explain the purpose of streak plate culture techniques, aka streaking for colony isolation. And 5, define the following terms: what's selective media, differential media, and reducing media. So the ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, meaning they dry out, or exposure to oxygen, like the anaerobes, and require a special transport medium to keep the specimen alive. Once a specimen is received in the lab for microbial culture, microbiologists employ the 5 principal I steps to process it. First, there's inoculation, then there's isolation, then there's incubation, then there's inspection, and then there's identification. All 5 of these steps begin with inoculation is placing the specimen on an artificial culture medium to initiate the growth of the specimen. 2, you then need to isolate the specimens so that you spatially separate microbial growth into distinct colonies. We'll talk a little bit more about this, but you need to do that before you can get a pure culture. And you really can't study a specimen, at least for clinical study, until you have a pure culture. Generally speaking, in the microbiology lab, we will be working with pure specimens. And that's so that you can do things with it and know that one species is causing the result. So that's isolation. Incubation is just placing the culture media in an environment to facilitate the growth of the microbes. Inspection is examining the culture for a presence of microbial growth. That begins to identify it, but generally to identify it, you then have to perform specific biochemical tests to ID the pathogenic microbes. Any question about any of that? And the Zoom file just completed, so my computer will now speed up. So the 5 steps discussed will be discussed further using different types of culture media. You're familiar with many types of media already, like you're familiar with general purpose media, such as nutrient agar or Na, or nutrient agar with yeast extract or NA, or TSA, or tryptic soy agar. These allow us to grow many bacteria, but not all bacteria, like fastidious organisms will not grow on a general purpose media. to promote the growth of fastidious microbes, those that have very specific nutritional requirements, we have to grow them on enriched media. And we usually call that enriched general purpose media. For example, we can add to tryptic soy agar 5% sheep's blood to help grow the specific microbe. And we don't call this tryptic soy agar with added 5% sheep's blood. We call that blood agar plates or BAP. Any question about any of that? And the blood supply the nutrients for many fastidious organisms to grow, like Streptococcus pyogenes or Neisseria gonorrhoeae, require blood agar plates to grow because they just don't grow on general purpose media. Any question about any of that? All right, so besides general purpose media, we have selective media. Selective media is a media that contains chemicals that inhibit or halt the growth of some organisms, but allow other organisms to grow. You should keep in mind when using selective media that first, the selection isn't necessarily perfect, meaning given enough time, even organisms that are selected against may grow on a selective media. And then second, even the organisms that are supposed to grow on selective media may form smaller colonies on the selective media than they would on a general purpose media. But the media does select for certain species to grow. Any questions about any of that? All right, we'll talk a little bit more about that in a little bit. There's also differential media, and this media contains chemicals that produce visible differences among different groups of organisms when they're grown on the media. These differences will be unrelated to how the organism grows on the media. Usually, different bacteria are visually distinguished from each other, meaning they'll have visual differences in differential media. And then lastly, there's reducing media. Reducing media promotes the growth of anaerobic bacteria because low or no oxygen conditions are required to grow these organisms. So we call an anaerobic media reducing media. And it's called reducing media because it has a chemical in it, which you don't need to know this, but it reduces to remove the oxygen from the media. It's important to note that a culture media can be classified into more than one category. For example, McConkey's agar is both selective and differential. So here we're looking at McConkey's agar here. It is selective in that gram-negative organisms can grow on McConkey's organism. Gram-positive organisms tend not to grow. Most gram-positives do not grow on McConkey's agar, but the gram-negatives do, so it is selective. It's selective because it has bile salts in it and crystal violet in it, and these don't allow gram-positive organisms to grow. You don't need to know why, but the crystal violet gets in the cell wall of the gram-positive and then stops the cell from growing. Bile salts are in the intestines, and a lot of gram-negatives grow in the intestines, so they're used to growing with bile salts. And gram-positives aren't used to growing around bile salts, so they don't grow with bile salts. All right, so that's why McConkey's is selective. McConkey's is also differential in that it allows you to differentiate lactose fermenters from non-fermenters. If the bacteria can grow on McConkey's and it ferments the sugar lactose, it will turn the plate and the growth pinkish or purplish color. And that's just because of the pH difference. When the bacteria ferments the sugar, it produces acids, which cause the pH indicator to turn pinkish or purplish. The bacteria that do not ferment the sugar lactose will not be pink or purplish. And this is a non-lactose fermenter over here. And that bacteria will be its normal color, whatever color that will be. Another media that's both differential and enriched is blood agar. It's an enriched media because it contains blood, which give nutrients for a fastidious organism to grow. So that's why it's an enriched media. And then it can be differential, allowing you to see 3 different types of hemolysis. Hemolysis is the ability to lyse the red blood cells. You can have gamma or no hemolysis. And here we have the white bacteria, and the red comes all the way up to the bacteria. So this is gamma hemolysis, no hemolysis. You can have alpha hemolysis, which is also partial hemolysis. And here you see the red comes up to a region which is darker. So this dark like shadow around the white alpha, which is the bacteria, that darkening is the partial hemolysis. And that usually looks either darker in color than the blood, or it looks somewhat greenish compared to the blood. And that is from partial hemolysis. Right there, we can see the partial hemolysis very clearly inside the alpha. It's a little bigger here than right here, where it's red. And then we can distinguish that from the complete hemolysis called beta hemolysis. The white B right here is the bacteria. And then this clear region, which is allowing the light to come through it, is complete hemolysis. And we call that beta hemolysis. So this is differential because we can differentiate 3 different species on their hemolytic activity. You'll note that whenever you have beta hemolysis, you usually have a ring of alpha hemolysis around it. And there's the alpha hemolysis, the dark ring around the beta hemolysis. This beta hemolysis gets rid of all the blood, so that if you were to lift up the plate and look through it, you could actually see through the plate right here. You can't see through the plate right here or where the bacteria is, but right here, you can see through the plate. Any question about any of that? All right. So this is showing you the different types of media and the different names. Let me remember if I mentioned everything. Yeah, I did. Okay. The different types of media. We have a general purpose media that includes TSA, NA, and NA, promoting the growth of many organisms that are not fastidious. We then have enriched media, and this promotes the growth of fastidious organisms. There are two plates which are enriched general purpose media, and that is TSA with sheep's blood, which we tend to call blood agar plates or BAP. And it is enriched, but it's also differential, allowing you to differentiate between 3 different species on their hemolytic activity. Another enriched general purpose media is chocolate agar or CAP. Chocolate agar is the same as blood agar, except that the blood cells are added when the agar is steaming hot. That lyses the blood, releasing their nutrients into the media, but that changes the color from red to brown. And because the agar has a brown color, they call it chocolate agar, or CAP for chocolate agar plates. Some organisms that do not lyse red blood cells have to grow on chocolate agar. And because they don't lyse the red blood cell, they don't get the nutrients from the blood. And one example, I'm only going to talk about one, is Haemophilus species. Tend to grow on chocolate agar, but not on blood agar. And we'll talk a little bit more about that later in the lab. For selective and differential media, we have McConkey's agar or MAC, eosine methylene blue agar or EMB agar, and heptone agar or HE agar. They're all 3 of them selective and differential media. They're all 3 the same, meaning they're selective for gram negatives, and then they're differential on their basis of lactose fermentation. Any question about any of that? And all 3 of these media that we're going to talk about just happen to be the same. Like mannitol salt agar is differential on another sugar, and we'll talk about that in a minute. Let's talk about PEA. It is a selective media, and this one is only selective. PEA is selective against gram negative, so we use PEA to grow gram positive bacteria. What happens is PEA has ethyl alcohol in the media, and ethyl alcohol gets inside the gram negative cell wall. It has lipids, so the alcohol gets in it and then stops the growth of the gram negative cell. The gram positive cells do not have lipids in their cell wall, and so they can grow on PEA. A mannitol salt agar is both selective and differential. It is selective because it has salt in it, 7% sodium chloride. Most bacteria cannot grow in 7% sodium chloride, and that's why it's selective. Only the bacteria that can grow in 7% sodium chloride will grow. It's also differential, that if the bacteria can grow on mannitol salt agar, then we can look to see if it can ferment the sugar mannitol. If it ferments the sugar mannitol, it'll produce an acid. You don't need to know that yet, but that'll change the pH to yellow. And if it's yellow, we then say this bacteria can grow on mannitol salt agar, which means it's a halophile, it likes the salts. And then if it's yellow, it ferments the sugar mannitol. If it can grow on mannitol salt agar, but it is not yellow, then it does not ferment the sugar mannitol. Any questions about any of that? I thought I had a picture of mannitol salt agar. Now, let me see if I can find it. It must be in another lab or in the worksheet because I'm not finding it. All right, inoculation is taking your media, in this case, media on a plate, and then inoculating it with the bacteria specimens. Usually at this point, if we have a clinical specimen, there would be many bacteria growing. And so we have to isolate it and then study a pure culture. And we'll talk about that in a little bit. But anyways, that's inoculation. You then isolate what you have growing. And Louis Pasteur first developed the concept of a pure culture. And what he did was he used limiting dilution of broth cultures. And so he'd make, several different dilutions, maybe 10 of them. And you would then take 10 replicas of each dilution. And if the bacteria grew in only one replica of that dilution, and 9 of the tubes were empty, Louis Pasteur said that very few cells started that 1 out of 10, because 9 out of the 10 didn't grow. So we probably have a pure culture. Most of the time he was correct. Not all the time, but most of the time he was correct that he did obtain a pure culture. However, this was a very difficult way to getting a pure culture. Robert Koch developed the streak plate method and made it easy to isolate colonies on an agar plate. And if you do that 2 times, you can get a pure culture. And this is much easier way to get a pure culture than Louis Pasteur's way of getting a pure culture. When you're streaking for colony isolation, you get your loop. Can you guys see this? We're going to pretend this pen is my loop. I'm not hearing anyone. Let's assume you can see it. You then put it in the Bacto incinerator and sterilize the metal or wire portion of the loop. Take it out, cool it, usually on the side of the dish that you have the stock culture in. Once it's cool, you pick up the stock culture and then streak it out on your agar plate, doing sector 1, shown right here. You then take the loop and put it in the Bacto incinerator and sterilizing it, killing the bacteria that's on it. And then you take it to your plate and you streak it out to form sector 2. Let me try and blow this up a little. What you do is your sterile loop, you go through part of sector 1 to form sector 2, meaning you pick up cells in sector 1 and then you streak out sector 2. You should only go back into sector 1 about 3 times. And they've done this a little too many times. That's 1, 2, well, it's only 3 times because the coming back is not picking up more cells it's going into. One problem here is that you don't want to come into sector 1 all the way where the bacteria are heavy, meaning in sector 1, the bacteria will be heaviest on this streak. And as you streak the bacteria out, the lightest bacteria will be on this streak here. So what you want to do is to set up sector 2 is you only pick up the bacteria where they're lower in number, meaning you streak it out in this region here. You don't go back into this region here. Anyone confused about that? All right. You then sterilize your loop in the Bactron incinerator, cool it, and streak out sector 3. Once again, you go through sector 2 to get the cells for sector 3. And what you should be doing is not coming all the way back where the bacteria are heaviest. That would be this region here. You go where the bacteria are light, and that would be this region here, that streak and that streak. So you go through there and here. And actually it looks like sector 4, they've done it correctly. And you get sector 3, and then you sterilize your loop. And then you set up sector 4, sterilizing the loop, and then coming in sector 3 to pick up cells. And you only do it where the bacteria are lower in number, meaning this region here. You don't come into this region here where the bacteria are higher in numbers. And then you set up sector 4. The goal is that someplace along the line, the bacteria will be diluted, and then we'll give you isolated colonies. Here we're seeing it in sector 4, but the isolated colonies could be in sector 3, or even in sector 2, or even in sector 1, if the bacteria are dilute enough to begin with. Okay? Any question about that? You then take from one isolated colony, and then you streak it out again on another plate to get isolated colonies. And the second time it forms isolated colonies, you then pick up the cells from cells that went through 2 colony forming events, like this colony here, which came from another isolated colony on the first streak plate. And then your chance of this being a pure cells, a pure culture, is very high. It's greater than 95%. Whereas if you were to pull them from here, the chance that that would be pure cells would be much less than 95%. Any question about that? So to get a pure culture, you send the cells through 2 colony forming events. And you're not guaranteed that it's pure, but it's greater than 95% chance that it is a pure culture. And then you set up your stock culture from this isolated colony that went through 2 colony isolation events. All right. Once you get your bacteria on the plate, you then incubate it, and that's simply put it in a place for the microbes to grow, meaning it has a physical environment to help the microbes grow. In our lab, our incubators are really primitive. All they do is they increase the temperature and maybe increase the humidity. If there's water in the bottom of our incubator, it increases the humidity. Now this incubator is much more fancy, and it can include changing the oxygen level, changing the carbon dioxide level, as well as the temperature and the humidity. And then you incubate it for a given period of time. Okay. Any questions about incubation. You usually incubate your cells in what we call an incubator. And here you're looking at an incubator right here. You then perform inspection on your colonial growth to see the different species. You may begin describing the colonial growth, like you can describe the appearance of the colonies, talk about the colony size, the shape, the margin, the elevation. That starts identifying the unknown bacteria, but it's not enough to identify it. To identify it, you then have to take a pure culture and run biochemical tests to identify the bacteria. All right, know the terms of this lab. And then you can skip the references. And then do the laboratory exercises. Let me go to the worksheet because it may be newer. Yep, it's about 4 months newer. All right, so answer the questions. There is no virtual lab for today. And that's unfortunate, but the virtual lab must have been using Adobe Flash. I always say it right there. So it's not going to run because Adobe is no longer supported. But we do have some videos for you to watch. How to inoculate an agar plate using the streak plate culture technique. You only have to watch that video to 4 minutes and 15 seconds. This plate only streaks 3 sectors instead of 4 shown in the lab module. Then watch a little video on beta hemolysis, alpha hemolysis, and gamma hemolysis. And then watch a little video on McConkey's agar. You only need to watch that to 3 minutes and 50 seconds. Then you can stop watching it. And then we have a little video discussing mannitol salt agar. So watch these videos and then perform the lab worksheet. Remember when you're uploading the file to only upload the worksheet. If you upload the lab manual, some of the questions may differ. And even if they don't differ, I'm going to slap on a penalty because the lab module is much bigger than the worksheet and it'll take up more space in my Canvas web space. And if I use too much space, I won't have any space to do future things. So I don't want to use up space. So please submit only the worksheet. Any question about any of that? All right. So in the worksheet, describe the natural circumstances where Staphylococcus species bacteria might benefit from salt tolerance. So you cannot say that Staphylococcus growing on mannitol salt agar benefits because it can grow, because mannitol salt agar is natural, not a natural circumstance. When Staphylococcus is growing naturally in the world, give me an example where it does, where it might benefit from tolerating salt. Okay, any question about any of that? All right, and then number 2, how might hemolysis benefit a hemolytic organism? The hemolytic organism lyses the red blood cell on the plate. How might that help the hemolytic organism? And then imagine you have a culture containing a single bacteria species. But the species may be one of several possible species. Describe a scenario where you use mannitol salt agar, blood agar, and EMB plates to help identify the unknown bacterial species. That is, how would you use these plates to help you identify the species? You should discuss two properties of each plate, like mannitol salt agar and blood agar, that you can use to help identify the bacteria. If you have less than two, you'll only get a partially correct answer. Any question about that? You will perhaps need a lot of space to write that down, but if you're writing in the worksheet, you can just hit the return key. What is the main goal for streaking for isolation? So state what the main goal for streaking for colony isolation is. Number 5, what happens to growth on the plate if you forget to sterilize the loop in between streaking the sectors for colony isolation? You need to be specific, discussing what happens with each sector, meaning discussing the growth in each sector. If you're not specific, you won't get full credit if you get any credit. And here's a hint. You should get a different answer for number 5. What happens if you forget to sterilize the loop in between sectors when streaking for colony isolation? And number 6, what happens to the growth on the plate if you transfer from stock culture to inoculate each sector? The growth on the plate will look different for number 5 than it will for number 6. So describe how the plate will look on each sector in 5 and 6. Any question about any of that? 5 and 6 aren't terribly difficult once you think it through, but you guys have not really worked with bacteria yet, so you'll need to think it through before you'll get the answers to 5 and 6. Any question about that? All right. Suppose you successfully streak a plate and obtain isolated colonies from 2 different bacterial species. That means you were starting with a mixed culture and you streaked for colony isolation on a plate. How can you use this plate to create a new pure culture of one of the bacteria? You must be specific in your answer so I can understand. All right, any questions about 7? In the table below, there are a variety of agar plate media available to microbiologists to cultivate bacteria. To note whether each medium is non-selective, meaning a general growth media, whether the media is selective, or whether the media is differential by placing an X in the appropriate column. The column may have, or the row may have more than one X in a column. You may use the table of media in the lab module to answer these questions. So we have nutrient agar. You have to state, is it non-selective media, a selective media, or a differential media? Or you could say it's all 3 or 2 of the 3. Whatever it is, put an X in it, and then move on. Tryptic soy agar. Is it non-selective, selective, or differential? Okay. And then 9, which plate here? I use the streak plate method to isolate single colonies. So A, B, or C. Let me briefly come back to the media. There was something I wanted to talk about with the media. I think I'll talk about it in the lecture. But when we're talking about different media, there are many media available to use in microbiology. If you were in the lab, I'd show you a book about this thick. So it's really thick. On every page of that book, there is one media. So that's a lot of pages of many different medias. So there's many different medias available to you. All right, any questions about any of the lab? If not, I will be here until 8 o'clock to answer questions. After 8 o'clock or until the last student leaves, I will leave. But I will be here until 8 o'clock or until the last student leaves. So go ahead and get working on the lab.
Thank you.
You're welcome.